e la relazione su tollerabilità dei preparati di immunoglobuline correlata alla metodologia produttiva. Volevo ricordare che il professor Faruggia è uno dei massimi esperti delle modalità di produzione delle preparati di immunoglobuline e in questo senso ha svolto una lunga attività in Australia proprio nella PPTA, cioè nella Plasma Protein Therapeutic Association, è consulente Kedrion e ci svolgerà questa relazione in inglese perché ha difficoltà nel parlare italiano. Prego, professor Faruggia.
Sì, grazie mille. Come dice, spiace che ho bisogno di fare la presentazione in inglese perché il mio italiano non è sufficiente. Spero che l'occasione migliora. Okay, thank you very much. This presentation is based on a paper written by my dear friend and very close collaborator, Professor Quinty, which is in the special issue of Frontiers in Immunology, which she referred to. I refer you to the issue. I'm sure that many of you are already familiar with it. It is a good collection of papers which we put together with experts in the whole field of immunoglobulin therapy. So it is my aim today to talk about the effects of manufacture, perhaps something which clinicians such as yourselves are less aware of. Now, this is a scheme of the classical cone fractionation alcohol system, which is used historically to manufacture the main plasma derivatives, including immunoglobulin and albumin. As I shall refer to later in the talk, and this has been alluded to by Professor Quinty, there have been variations in this scheme over the years. The point I want to make immediately is that there are a number of important parameters which are variables in generating the products in this scheme, including the concentration of alcohol, the temperature, the pH, the protein concentration, and so on. So there are a large number of things which are changed over the course of fractionation to give us the products. And the variation, the variability possible in these parameters has a marked effect on the product. For example, looking at this particular step in the manufacture of confractionated immunoglobulins, such as IgG, you can see that the variation of just 0.3 points in pH results in a difference in stability, so that we get a fragmented molecule if we drop the pH from the optimal, which is 5.4. So the point is that we can get very different products in terms of tolerability, safety, and efficacy based upon the variation in the manufacture. This is extremely important because, as has been already pointed out by Isabella, immunoglobulin products cannot be considered to be generic precisely because of this reason. The variations in the manufacturing process, which are controlled to a large extent by manufacturers, but which are always going to be subject to change, result in different products. In fact, the way we express this in the field is the product is the process is the product. And this results very much, this variation in products, this difference between products, in differences in tolerability, which I think you are very familiar with, but it can also result on relatively rare occasions in differences in efficacy. And this is a paper published by a group in Taiwan who have published on this subject twice. And you can see that comparing the efficacy of immunoglobulin intravenous in Kawasaki disease, we have 4 different products here. This particular product here, in fact, has a lower level of efficacy, and this is significant. And this is related to some particular features of the manufacture of this product. So I think that the point is very well taken. So now what I'm going to do is to go over as much as I can on 3 particular problems which have faced us in this area over the past 25 years, in which problems and unexpected adverse events occurred, and we know now as a result of unpredictable effects from variations in some shape or form of the manufacturing process. Going to start with the very well-known epidemic of the transmission of hepatitis C. And many people in this room remember this well, including some of them, including Professor Quinty, who has worked extensively on this subject. You know very well that although with some sporadic exceptions, of non-A/non-B hepatitis following early IgG preparations, there was a long history of safety from an intramuscularly administered immunoglobulin, and this showed us that it was not necessarily the case that this was going to be the same with the intravenous preparations. Nevertheless, despite the fact that this could have been to some extent predicted, viral inactivation for immunoglobulin products was considerably delayed relative to the coagulation factors used for hemophilia. There was a belief, based on relatively little science, that confractionation cleared the virus away from the products. You will remember that about 22 or just over 20 years ago, several hundred patients on both sides of the Atlantic developed non-A/non-B hepatitis. In fact, this was already characterizable as hepatitis C by one particular product, which at that time was GammaGard from Baxter. Some interesting investigations showed results after this outbreak. First of all, this is a study published by the FDA laboratories before the outbreak actually occurred. In this study, plasma which was known to be positive for antihepatitis C was fractionated in a small scale in the laboratories of the FDA, and the immunoglobulin fractions over the course of the fractionation by cone, as you see here, were tested using RNA PCR, detection of RNA in the fractions. The point I want to make is that in fraction 13 here, the sample 13, which was the actual immunoglobulin product, there was HCV RNA. Now, the point I want to make is this. There is no question that even though subsequently we introduced the test for antihepatitis C in all the plasma donations, every single pool was contaminated with hepatitis C before this happened. There's no question, because the epidemiology of the disease was such that we knew that because of the large-scale pooling that we had hepatitis C. Despite this, most hepatitis C, most immunoglobulin products were totally safe from hepatitis C. Now, what happened? In the intervening period, therefore, the hepatitis C antibody test was introduced. This is the structure of the genome of the virus and the various proteins which are expressed. And you know that we had various generations of testing. The first generation, the first antibody test, which was using for the study I've previously shown, had a specificity against the C and the first envelope proteins, and subsequently, very rapidly, different generations of tests were introduced which were more sensitive. However, very remarkably, when the GammaGard incident occurred, the FDA laboratories showed that the introduction of the second generation test ironically resulted in the finding that hepatitis C RNA was more present in these batches, despite the fact that there was on the test, that there was the test, and that in fact the recipients of these batches which were made screening with the second generation test, as you can see on this slide from the paper published in Lancet, were the ones who developed hepatitis C. So we think that, and on the basis of other experiments which I'm not showing here, that what happened when we introduced the second generation test for the virus, that the antibodies which were complexing with the virus to clear the virus away from the product were absent, and therefore the virus ended up in the product. This was totally unpredictable, very unexpected, but it shows us how a change in manufacture, any kind of change, including testing of the product and testing of the plasma, can result in unpredictable events. Another manufacturing type incident, also unfortunately involving hepatitis C, occurred with a product which historically had never given any problems, and this was hepatitis C through anti-D. And there were two separate large-scale epidemics, one in the Republic of Ireland and one in East Germany, as it was then before the removal of the Iron Curtain. And in both of these epidemics, which you can see summarized here, a large number of women, very tragically, were infected with this virus. Much of the infection was detected very retrospectively after the event. Why did this happen? This happened again because of manufacture. In both these countries, there was a great eagerness to produce anti-D immunoglobulin domestically and not imported from other countries. To do this and to get self-sufficiency in order to have enough immunoglobulin, the manufacturing was changed from the cone fractionation system to a purely chromatographic method. This, I emphasize, occurred before the products were in the era of viral inactivation. Therefore, any protective effect, and we know that the protective effect was not full, from cone fractionation was removed, and very tragically, one or two donors got into the pool who had hepatitis C and proceeded to infect the product, and large numbers of women. This is one of the worst catastrophes in the safety of blood products and was investigated thoroughly by an investigation commissioned by the Irish government. This is a final slide in relation to this issue, and I just want you to focus here on this data, which is showing us what we call model virus removal, in which viruses known to be related to the viruses of interest are studied in small-scale models of the fractionation. And the point I seek to make here is that what we've got is a study showing fraction 3 removal, which is what happens during immunoglobulin before we get the fraction 2, which is therapeutic. And you can see that for many of these model viruses, we see a large log removal during this fraction 3 removal, which is a very good thing because this is removing viruses from the immunoglobulin. But for the bovine viral diarrhea virus, which is the model for hepatitis C, we do not see this removal. And so we come again to the conclusion that the cone fractionation on itself is not enough to make the immunoglobulin product safe. And this is why these days, 20 years after these bad events, we have viral activation. I want to now move closer to our era and talk about the well-known phenomenon, which I think many of you are familiar with, of thrombotic complications. And you know that, again, somewhat unexpectedly, about 4 or 5 years ago, the Octagam product in particular was pulled out of the market on both sides of the Atlantic by all the regulatory authorities because of a higher than expected risk of thromboembolic events. Why do I say higher than expected? Because thromboembolic events, as you know, have been one of the known very rare adverse events coming from immunoglobulin. All of a sudden, 5 years ago, for this particular product, as it was thought at the time, this was not very rare, and the product had to be withdrawn. And then some subsequent studies started to be made by the regulatory authorities. And it is interesting to see, for example, this is the paper from the FDA in the United States, which shows the risk of the various products they studied, but observe that it was not only immunoglobulin from Octagam which had a higher risk. There were other products, including, surprisingly, this then in the market subcutaneous product from another company. And it is interesting that around about the time that this started to be detected, this product was also quietly, not as dramatically as Octagam, withdrawn from the market. So it wasn't just Octagam, it is interesting. And this is the equivalent data set from Europe, from the Paul Ehrlich Institute. You can see here various products over here, and you can see the way in which the incidence of thromboembolic events suddenly increased dramatically in the case of Octagam in these years, but you can also see a significant increase in this particular subcutaneous product, which, as I said, was very quickly withdrawn from the market and succeeded by another product. Now, why did this happen? Again, let us look at the manufacturer. This, I draw your attention again, is cone fractionation is the classical scheme. It is the way it was developed to a greater or lesser extent initially, and you can see that as we get to the immunoglobulin fraction, the prothrombin is not included. When we say prothrombin, we mean procoagulants, because at this time, in Cohn's time, many of the coagulation factors were not discovered, and it is going further down. So it doesn't go into the fraction which we make immunoglobulin. However, variations of the scheme have existed for many years. And we know these days that the so-called Kissler-Nishman variation of cone scheme, which is still ethanol fractionation, but which is actually very different in the way the various parameters which I have told you about, pH, protein, ethanol, and so on, are manipulated, it results in a different fractionation scheme. And we know that the precipitates from which the immunoglobulin is harvested are very different in composition. And this is a quotation which I think is very significant from the Swiss fractionators, Kissler and Nishman, which says that slight deviations in ionic strength will influence yield and purity very markedly. We think that this is definitely what happened in the incidents I have described, in which fractionators, companies attempted to improve yields by varying the conditions. It's very easy to do this, but it's not very easy to predict what will happen. And we know that as a result of this, the content of factor 11 and the immunoglobulin increased markedly, because factor 11 actually always has a tendency to co-precipitate with immunoglobulin. It's not surprising that this happened. And in fact, when this is another publication from the FDA, when you look at the factor 11 content through thrombin generation, you can see that certain products have high content. And if we look and this particular slide, I think this is more clear. This is the coagulation time, which is used to measure the activation. You can see here different batches of octagram. The first batches which were involved in the thromboembolic events. Notice how short the clotting time is. This means there's a lot of activated clotting factors. Then this is the old process, but no thromboembolic events. And then the process was modified in order to remove the coagulation, activated coagulation factors. And you can see that we get a nice prolongation of the coagulation time, indicating higher levels of safety. Observe now, though, that we have the globulin process again. This shows us a high level of activated coagulation factors in the old process, but when changed to the new product, this becomes a much better situation. So again, we emphasize a change in manufacture introduced for very good intentions to improve the provision, increase the yield, resulted in an increase in adverse events. We are now very satisfied that the products are safe. This is one particular product which has been published well. This is, I believe, Sibogama from Grifols, and you can see that over the course of the fractionation, the amount of factor 11, which is the guilty protein in this instance, is going down to limits which we cannot detect into the final product. So this is very satisfactory. And I now come to the last adverse event, which has already been discussed somewhat by Professor Quinty, so I will be able to go over some of these slides in more speed, and this is the question of hemolysis. This is a publication from the United States, from an FDA workshop done last year, and it shows that, again, this extremely previously rare adverse event in certain products has increased over the years. It's very curious that this started to happen. Again, this is exactly like the thrombotic situation. We have had on the product information sheets pointers that hemolysis is possible with IVIG, but it was a very rare event. All of a sudden, some products started to show not so rare an event. And when these investigators who surveyed the products, which you can see here, studied the manufacture, they found that they could classify the risk posed by the products into two groups. This is published in Transfusion. I draw your attention to the paper. And one group in particular showed 95% of the incidence of hemolytic events, whereas the other group only had about or less than 5%. And the difference between these two groups was, in general, a difference in the method of manufacture. In the group which showed very little hemolytic events, as is historically the case, we had a fairly straightforward progression of the cone precipitation system with alcohol, whereas with the group which showed the majority of the hemolytic events, we had a deviation in which, in order to achieve higher levels of yield and purity, we got a chromatographic step. This is shown in this slide, and you can see here another investigation on a particular product. This is the Prevision product as it was then, and you can see that compared to confractionated IgG, the product had high levels of anti-A isoagglutinin, which is thought to be the causative event, the causative entity in the hemolytic event. Isabella has already described for you the mainstream introduction of a step to prevent this by the industry, and you can see that when this particular company introduced immunoaffinity chromatography to remove the isoagglutinin through binding through the relevant blood group substance, this led to a very low level of isoagglutinin binding. And therefore I come to the conclusion, and I hope I have not said too much, or that I'm not in excess of my time, but I think we can be satisfied that these treatments over the years have enjoyed a high level of safety and tolerability, but we have seen some hiatuses, which I have described to you, and this happened when the manufacturing method was modified without sufficient consideration. I think it is very important to realize that when the manufacturing method is changed, this has to be approved by regulatory authorities. We know this. But the regulatory authorities do not have the capacity to detect adverse events through the very small clinical trials which are used to gain pre-market authorization. It is only possible to gain a sufficiently large population of patients to study post-market. This is a very important thing for people to realize. There is a difference between efficacy and effectiveness, and this is the study of effectiveness. It's very important to understand that we are only able to detect adverse events after approval, which is a big deficiency in the system. Hence, we must be very cautious as manufacturers and as prescribing clinicians when considering any manufacturing change. But I want to finish my remarks by emphasizing that these days, following these accidents, IG products continue to be extremely safe. Grazie mille per l'attenzione.
Bene, possiamo fare brevissime domande con brevissime risposte al professor Spadaro e al professor Farrugia, visto che la professoressa Quinti deve andare via per motivi